Today we'll be going over Chapter 4 on the three-dimensional structure of proteins. Alright, we're looking at a peptide bond right here, and proteins are made up of amino acids, and when two or more amino acids condense together, you get a peptide bond. And the peptide bond is going to be kind of important for this chapter. And you can see here I highlighted this CN portion, and it turns out that it looks like this is only a single bond, but because of resonance between this carbonyl oxygen and this nitrogen, there's a partial double bond character to the peptide bond. And so this carbon-nitrogen bond does not rotate. There's no rotation about the CN bond, about the peptide bond, and it's double bond-like and it's planar. Alright, and I should maybe just go back and kind of orient you to what's happening here. And so this alpha carbon, that's the central carbon in the amino acid, and then you've got the amide right there, and then this is a different amino acid right here. This is the central carbon with the carbonyl group, and the carboxyl and the amide group are going to condense together to form that peptide bond. So this is the peptide bond right here, and there's going to be no rotation there because it's partial. It's like a double bond kind of. And this NC bond is called a phi bond, and then this CC bond is called a psi bond. And the phi and psi bonds can rotate, but the peptide bond cannot rotate. Alright, we're going to get into some secondary structures. There's three types we're going to talk about: alpha helix, beta sheets, and beta turns. And we've talked about four types of weak bonding that occur in proteins, but in the secondary structure, all we're going to see is hydrogen bonding. Okay, so we'll first talk about the alpha helix. And if you look right here, there's a pole going down, and then this alpha helix is kind of rotating around that pole. The pole is not part of the alpha helix, it's just kind of a schematic diagram. And so if you start here and you take one complete turn, then you're going to use 3.6 residues, or 3.6 amino acids, and it's going to be 5.4 angstroms in length. And so 5.4 angstroms equals 3.6 amino acids. And so you could be asked questions on the test such as, how long is a polypeptide with 80 amino acids? So you'll do some dimensional analysis. So you'll take 80 amino acids, that's on top, and so you want amino acids to be on the bottom when you're multiplying. So 3.6 amino acids would go down here, and then on top you'd have 5.4 angstroms. So you take 80 times 5.4 divided by 3.6. And then in alpha helices, the hydrogen bonds are going to be internal, and so the hydrogen bonds are occurring inside the structure, but the R groups are external, and so these purple balls represent the R group, and you can see that they're all sticking out externally. And then this diagram is just showing that you can have different polarity to different sides, and so like this yellow part, I can't remember exactly what it represents. It's either polar or nonpolar, but one side can be nonpolar and the other side can be polar, depending on the orientation of the R groups. Okay, alpha helices can be left-handed or right-handed, and this diagram is just showing how you tell if an alpha helix is left-handed or right-handed. I don't think you guys will ever have to worry about that. But if the way that you do it is you make your you just put your hand like that, and then the fingers right here are pointing in this direction, and so that's the direction that the alpha helix will move. And so it moves in towards the right, and your fingers are pointed towards the right. Over here your fingers are pointed towards the left, and so it moves towards the left. Alright, and so we have a bunch of amino acids listed right here, and it turns out that alpha helices only have certain amino acids. And there's some numbers right here, and the lower the number, the more likely that amino acid is to be found in an alpha helix, and the larger the number, the less likely that it will be in the alpha helix. And so alpha helices have lots of alanines and very few glycine and proline, and that's because of sterics. Proline, if you think about proline, it's got that strange kind of ring structure on the R group, and so sterics would prevent it from forming that alpha helix. And then alpha helices have a dipole associated with them, and that's because of the orientation of the groups. And so at this end you'll have a lot of amino termini, and so you'll have positive, and down here you've got the carboxyl terminus, and so it's going to be negatively charged, slightly negatively charged. Okay, so just as a review for the alpha helix, 5.4 angstroms is equivalent to 3.6 amino acids, and that equals 1 turn. The hydrogen bonding is internal, and the R groups are external. Alpha helices have lots of alanine, very few glycine and proline, and they have a dipole. And then we'll see that keratin is made up of alpha helix. Okay, we've got beta sheets next. And so we're going to talk about how beta sheets can be anti-parallel or parallel. And anti-parallel is more stable, parallel is less stable. Just like the alpha helices had a certain type of amino acid, beta sheets also have a certain type of amino acid, and in this case they're going to be small. The R groups are going to be small. And then the R groups alternate up and down. And so this is the first type, anti-parallel. And here's a top view, and then here's a side view. So it kind of zigzags. And this anti-parallel is more stable because of linear hydrogen bonding. So right here we have linear hydrogen bonding, and so these three atoms are in a straight line. And then note the R groups are alternating up and down, up, down, up, down. And then this one is parallel, and in a parallel beta sheet the hydrogen bonding is bent, and so that's going to be less stable than the anti-parallel. The third type are beta turns, and on the beta sheets they kind of move this way, and then they kind of curve around, and that curving portion is called a beta turn. And there's two types, type 1 and type 2, and the types are dependent upon the amino acid in the turn. And so in type 1 we have proline, which has that, remember that circular R group, and that anytime you see a proline it's going to cause like a kink, and so the proline really helps to cause that turn, that kink in the beta turn. And then type 2 we've got glycine, and type 1 is going to be more common than type 2. Alright, and we're going to talk about some fibrous proteins, and there's proteins can be categorized in a couple ways. You've got fibrous, and then you've got globular, and globular are going to be like really folded on top of each other, and just kind of like the name says, is they're going to be globular. Fibrous are going to be more straight. And there's three types of fibrous proteins that we're going to talk about: keratin, fibroin, and collagen. And this is a good table to just kind of look through. Maybe pause the video and check it out. I've got a table at the end that kind of sums up a lot of it. I'm going to compare some characteristics of keratin and collagen. Alright, here's keratin, and I'll just skip kind of to the table just to show you the categories that we're going to look at so we can know what we're comparing. So for keratin and collagen, we want to compare handedness, what's going to cross-link them together, their secondary structures, and where they are found. And this isn't like a comprehensive list, but oftentimes the teachers like to ask like for comparisons and contrasting of the keratin and collagen. So here's a keratin. Keratin is an alpha helix, and it's right-handed when it's an alpha helix. Then two of these alpha helices combine to form something called a coiled coil, and when it becomes a coiled coil, it's left-handed. Now, just to contrast the collagen, collagen is not made of an alpha helix, it's made of an alpha chain, and then it starts left-handed and becomes right-handed. Okay, and then you take these coiled coils and you just kind of join them together into these groups, protofilaments, and then you stack protofilaments on top of each other to form protofibrils. And these protofilaments are cross-linked by disulfide bonds, and so I drew these little black things in here. And so these disulfide bonds help to join, help to kind of stabilize the keratin. And collagen is going to be cross-linked by something different. So here's a picture of some hair. So we find keratin in hair. This is one molecule, one piece of hair. And you start with the alpha helix, and it's really just that that little ribbon that's going through there. They just kind of draw this box thing around it just so that it makes it easier to visualize. And then it becomes a coiled coil, and it becomes a protofilament and a protofibril. And I wouldn't get too hung up on these structural terms. I wouldn't worry too much about those. And here's collagen, and it's an alpha chain. Starts left-handed, goes right-handed. It's cross-linked by something called high lyse. It's kind of a modified form of lysine. And it kind of looks similar, but it's you got the groups aligned, kind of offset by each other, and they're cross-linked. There's 3 alpha chains, whereas there's 2 alpha helices in keratin. And so here's just a comparison. Just maybe pause it and look at the table. Okay, we're going to talk about perms. Like a perm is like when a lady goes to get her hair curled at the salon. And so here this kind of represents like the hair, and you've got these disulfide bonds. And remember, when you reduce, it breaks those bonds. And so right up here I've got reduce, and then you twist the hair, and then you oxidize it to reform those disulfide bonds, and that gives you a permanent curl. And so it's kind of like a random slide, but that's kind of like I guess a biochemical application for perms. And then you've got fibroin, and fibroin are going to be made of beta sheets. And so what kind of amino acids would you expect to see in fibroin? So you'd see small hydrophobics. And yeah, I don't think you're going to be tested too heavily on fibroin, but it's kind of like silk, and it's made of beta sheets.